Nanosene is the first bespoke polymer supplier from Africa, designing and supplying polymers intended for molecular drug target identification. For researchers and industry to design and discover new therapeutics, it's essential to study and understand these intricate membrane protein drug targets.
And here's why. Membrane proteins facilitate a range of fundamental biological processes, such as the flow of molecules through and across membranes, responses to chemical messengers, and cell-to-cell communication within the human body. Numerous diseases such as asthma, obesity, high blood pressure, Alzheimer's, and cancer are associated with membrane proteins, thus making them important targets for drug discovery and development, as well as intriguing research subjects. Clearly, membrane proteins are crucial in effective drug design, so why do we know so little about them? The problem stems from the difficulty in separating or isolating them from the cell membrane to be able to study their structure and function. The native environment or cell membrane in which these membrane proteins are embedded consists of two layers of phospholipids, each with the hydrophilic head and hydrophobic tails. Membrane proteins mimic the amphipathic properties of the phospholipid bilayer by having both a hydrophobic part embedded within the membrane and a hydrophilic part on the outside of the membrane, which is in contact with the surrounding aqueous environment on the inside and/or outside of the cell. In contrast to soluble proteins, the amphipathic properties of the membrane proteins prevent solubilization in standard aqueous buffers, as the hydrophobic part of the membrane protein bundles together to minimize contact with the aqueous environment. This undeniably changes the structure and folding of the membrane protein, typically hindering studies of its native form. This problem can be addressed using synthetic polymers to form polymer-bound nanodiscs. Water-soluble amphipathic polymers are able to interact with and insert into the cell membrane to isolate and stabilize membrane proteins with their surrounding lipids in nanoscale discs. These nanodiscs enable the analysis of membrane proteins in a native-like environment while retaining their physiological properties. This technology offers researchers and drug developers the unprecedented opportunity to access some of the most valued proteins in a biologically active state.
Nanosene's technology facilitates more successful identification of drug targets through the design and synthesis of new functional polymers that can outperform conventional methods. The controlled synthetic strategy that we employ is highly customizable, allowing for dedicated polymer design and integration into current workflow methodologies and various specialized applications.
Nanosene has developed new amphiphilic copolymers to address limitations of currently available polymers. Benefits of these new polymers include predetermined molecular weights with narrow chain length distribution and the possibility to precisely tune their hydrophilic-hydrophobic balance. Compared to current materials, our material offers the possibility to introduce additional functionalities such as fluorescent labels, affinity tags, and the tethering to surfaces.